Salamander Courtship. 2 weeks after Dick and I conclude our expedition to Costa Rica, I am sitting in Peter Marlar's animal behavior course at UC Berkeley. It is late September 1965. Marlar is a charismatic lecturer. His classroom is packed with 50 undergraduates and graduate students. I have high expectations for the course, and I am not disappointed. By the end of the semester, I decide that I will study the evolution of behavior for my doctoral dissertation. I will take the approach of comparative ethology, which is to discover evolutionary change in behavior by comparing related species. Marlar's course is a delight and a challenge. He lectures from notes that will become a leading textbook when it is published a few years later, but that is in the future. Instead of a textbook in sync with the lectures, we have a book of Readings in Animal Behavior by Thomas McGill, a collection of fascinating case studies. Marlar is original and brilliant. His lectures sparkle with new ideas and overarching concepts: stimulus filtering, temporal patterning, reciprocal inhibition, and critical periods, to name a few. We must write a term paper for the course. Marlar hands out a mimeograph list of possible topics. We are instructed to look it over and then make an appointment with his secretary so that we can meet with Marlar himself to make a final choice of topic. At the appointed time, I huddle with Marlar and come away with my topic: chemical communication in animals with a focus on pheromones, chemical signals. Marlar also gives me a reprint of a recently published article by Keith Nelson, one of his many graduate students. Nelson describes the courtship displays of related species of fish that deliver pheromones to sexual partners. He uses Markov chain models to analyze the chaotic sequences of behavior that unfold during courtship. A few years later, I used the paper as a blueprint for one section of my own doctoral dissertation. The next semester, Marlar gives me one additional gift. I am enrolled in an honors course called Pre-Seminar. In this course, the instructor, congenial Professor Nandi, pairs each student with an advisor who will help them choose a scientific paper. Then, with the advisor's help, the student prepares and delivers a talk on the paper. The talk is given in front of the pre-seminar class with the advisor present. Nandi pairs me with Marlar, who suggests that I give a talk on a recently published paper on pheromones. It is my first experience at public speaking. My talk is a success. Marlar shakes my hands, and, like the crack of a champagne bottle on the bow of a ship, my scientific career is launched. In my senior year at Berkeley, I qualify as an honor student. That achievement means that I need to research and write a senior thesis. I decide to do my senior thesis on the mating behavior of Hymenocyrus betgeri, a small African frog that I have been breeding in the basement of the Jones Stanley Tropical Fish Store, where I work part-time. But a quick trip to the library reveals that their behavior has just been described by George and Mary Rabb at the Brookfield Zoo in Chicago. Then I remember Robert Stevenson's recent lecture on the courtship behavior of a local plethodontid salamander, Ensatina eschscholtzia. Ensatina behavior is both bizarre and complex, but courtship hasn't been observed in any of the other plethodontid species that live in Northern California. I float the idea of a comparative study past Stebbins. He readily agrees to sponsor my research and recounts the details of his success with Ensatina in 1949. Stevenson's trick is to find mature and sexually active salamanders in the field and bring them into the laboratory so that courtship can be staged and observed. Stebbins sets up a terrarium on a table next to his bed, covers it with a diaper, and illuminates it with dim red light. He wakes up periodically during the night and checks for courtship. Instead, I set up terraria in a small refrigerated corkroom next to the herpetology lab of the museum. Now all I have to do is obtain live salamanders for my project. Dick Sage and Ross Keister agree to help me collect salamanders over a long weekend in February 1966. Our plan is to head north from Berkeley to Humboldt County, where we hope to collect plethodon, and then east to Shasta County, where we hope to collect hydromantes. Night falls before we reach our first objective, so we pull off the road, set up our tent, and crawl into our sleeping bags. Ross wakes us in the night with a plaintive wail. My sleeping bag is soaked. I mean, completely soaked. In contrast to Ross's predicament, Dick and I find that our sleeping bags are completely dry. A puddle of rainwater occupies only one corner of the tent, and Ross's sleeping bag does an excellent job of wicking it up. Dick and I go back to sleep while Ross tries to make himself comfortable in the car. Over the next few days, we have limited success finding plethodon and no success finding hydromantes. We find a pair of ensatina under a rock near Mount Shasta. Later, these animals court in the laboratory and give me crucial data on ensatina courtship. I have limited success with my senior thesis project despite hours spent at night in the corkroom. I try to observe courtship in several species of plethodonid salamanders, but only succeed with ensatina. But even that limited success is enough for a thesis. No one has seen ensatina courtship since Stebbins observed it in 1949. I produce detailed accounts of several hours of courtship and compare my results with published accounts of other species. A few years later, at the University of Michigan, my fellow graduate student Steve Tilley makes a huge contribution to my plan to do my doctoral research on salamander courtship. For his own thesis, Steve explores reproductive investment, a hot topic at the time, by documenting size-fecundity relationships in 5 species of desmognathous salamanders in the Appalachians. In the course of that work, Steve helps me establish the feasibility of my developing plans for a doctoral dissertation by supplying live animals at a crucial juncture. In the summer of 1967, Steve and his wife Mary returned from North Carolina with a gift for me, a sample of mature, sexually active, red-legged salamanders, Plethodon shermani. I soon find that, in contrast to ensatina and the other West Coast plethodontids, eastern plethodon courtship is easy to observe, photograph, and film. My plans for doctoral research begin to form around this success with plethodon. Over the next 50 years, I work on other projects, but retain my interest in the sex life of salamanders. I publish a couple dozen top papers on this topic. Along the way, I commonly introduce myself to new dinner companions as the world's expert on salamander courtship. A few of my professional colleagues think I'm foolish and even reckless to lavish so much effort on such a frivolous topic. What do they know? I have a reason for my obsession. In the course of my doctoral work, I discovered that some parts of salamander courtship sequences are more variable than others. In particular, the part dealing with sperm transfer is stereotyped among species and genera, even though the earlier parts of courtship are highly variable. Why should this be so? By the end of my career, I have an answer to this question. Sperm transfer involves intricate interaction between male and female. Other parts of courtship also involve interactions, but the consequences of deviating from a strict back and forth sequence are less severe. The couple may be out of step during preliminary courtship, but sperm transfer does not fail as a consequence. In other words, the premium on precise sexual interaction during sperm transfer is higher than in earlier parts of courtship. This difference in premium is captured by the concept of multivariate stabilizing selection. This mode of selection is the key to understanding why particular modes of sperm transfer are conserved over hundreds of millions of years in salamanders. Strong stabilizing selections means that even small deviations from the optimum combination of male and female traits are not tolerated. Furthermore, the position of the optimum itself is locked in place by the interactions. The same idea of evolutionary conservatism applies to all other kinds of characteristics that involve interaction within and between individuals and strong stabilizing selection. My obsession with salamander courtship helped me discover, articulate, and model an evolutionary concept of broad significance.